Okay, day 16. Oh no, day 17. 17 days. And today we're gonna go back to DNA. Deep dive. What happening? What is this? Some kind of AI? Winter stack for the fish. Mm-hmm. We don't need that, we don't need that. Refresh. Okay, let's go. So, counter impact vision, yeah. That's good. Let's start a new chat. Project. Okay, please tell me or explain me like I'm 14 years old the process of cellular biology, that's reading DNA, converting it into an array or mRNA, and that then mRNA converting it to the protein. So the last part I understand, but first part is confusing. Who is reading the small chunk of DNA to create mRNA? Who is doing that particular? And also, how does it know where to start on that long DNA chain? It's gonna be interesting. Let's go. Let's dive into it. Also, where's my grok? I need to open a grok. That's my grok. YT. Again. Yep. I think it's for so long. 4.7. Adaptive. Taking its sweet time. Okay, great question. Classic. Classic so far. Long dash. That's what's AI doing. That's the best. AI really is the best at long dashes. This is one of the genuinely confusing part of biology because there is no single neat answer, and most explanation skip the who and how entirely. Exactly. That's why I'm confused. Like I don't know who doing the reading. The molecular machines that read nDNA and produce mRNA, an enzyme called RNA. Polymerized 2. RNA Pol 2 is a good YouTube videos. Please give me links or give me search. Couldn't read it, and most importantly, I didn't wanna bore you to death with this information. Let's see if I understand first. Who's doing that? The model molecular polymerase R2 RNA Pol2 for short. RNA Pol2. It's a big protein complex. Picture something like a tiny tape head that physically clamps onto DNA and zips small sections. That's the key. How does it know which section? Slides along and synthesize mRNA letter by letter using one of the two DNA strands as a template. By the way, is it same RNA happen from different sides of DNA? Probably not, right? And that's another deep dive that we're gonna do that. You mentioned it's originated from one side of DNA, one strands, right? So mRNA will be completely different or symmetrically opposite if RNA Pol2 reads the other strands, right? So now we have a questions. Who is doing it? We kind of already know. It's RNA Pol2. We still don't know why it starts in that particular location. And also, how does it pick that strand and not the other? Because I'm assuming results would be symmetrical but opposite. Clip that. Oh, by the way, here's the YouTube videos. Some really good stuff out there. Let's go. I need to understand visually, but before that, templates, that's really what's happening just at the molecular scale. It grabs the tape, unwinds it, reads it, and bumps. Reads the bumps, write a copy onto new ribbon as it moves. Totally makes sense. Just my questions. How does it know where to start? Here's a thing. RNA Pol2 on its own, it's pretty dumb. It cannot recognize this is the start of a gene by itself. It needs help. Just before each gene, there is stretched of DNA called a promoter. Oh, okay. So promoters. The promoter is, I need to start writing that. So machine itself, it's a, I'm gonna use my iPad to study. Okay, so we open up the sketch. Oh no, my pen is dead. Is it? Yeah. So we have what do we have? We have RNA pol2, RNA pol2, and then we have promoter. Promoter. Here the things RNA pol2 its own, it's pretty dumb, yeah, you're right. The promoter isn't part of the gene itself, it's a start here signal written into the DNA. Again, yeah, I understand the promoters, but I'm assuming there is thousands of different promoters, right? Oh, how many genes do we have? That many promoters, right? The question is, how does it know that it should start from this promoter? So we probably have promoter before each gene, and probably more promoters than just a gene. So if I remember correctly, we have 23 chromosome, so 46 total, and each chromosome has thousands of genes, right? So I'm assuming there's thousand upon thousands promoters. How does it know on which promoter to start? And don't tell me RNA, because RNA tells what's not available to read. So it definitely blocked some promoters to be started there. But from thousands of others, how does it know that start from number 3 or 4? I'm leading actually to one of the thought I had earlier. So I need to understand the process to see if my thought even valid. Oh great, I didn't reply it. So I'm assuming there is thousands of promoters in DNA. How does it know at what promoter to start? And don't tell me RNA, because RNA just make some genes unavailable to read. So there is still many options to start. How do we know at which promoter it will start? I need branching. Like this is not linear, doesn't make sense because I'm gonna repeat myself shortly, right? Isn't part of the most famous landmark. Clip that. The most famous landmark inside memory promoter is a sequence called the Tata box. Ta-ta-ta. A strength of DNA that reads ta-ta-ish, sitting about 25-30 letters upstream of where transcription actually begins. Totally makes sense. A separate group of proteins called general transcription factors binds to the promoter. First, the key one, TBP, or TATA binding protein, specifically recognize the TATA box and bends the DNA. There. Other transcription factors stack on top, forming a scaffolding called the pre-initiation complex. Only then does RNA pol2 get recruited and dock on that scaffold. Okay, so they built some kind of transport, like platform where the RNA will do. So promoter itself Tata binding protein. Okay. The platform called Tata binding protein or TBP. Okay. Uh, and that's it. Tata. Ta, ta. Okay. A separate group of proteins called general transcription factors bind to the promoter first. The key one. Yeah, I already read that. In your terms, the genome is like a huge binary blob. Promoters are magic bytes, headers that mark. Yeah, exactly what I'm thinking. Executable regions starts here. Transcription factors are the loader that recognize the header, format, and hands control to the actual interpreter, RNA-POL2. The polymerized the polymerase doesn't parse the file format itself. It just executes once something else has positioned it. Okay, something else. See what is this? A gap in science? That's what we see here? I'm just trolling. Why and click that. Why all this machinery for one job? The separation matter. It's how the cell control which genes get expressed and when. Yeah. Different transcription factors are present in different cell types. Oh, yeah. That's confirming my thoughts. That's very good. And in response to different signals. So the same gene can be on a liver cell and off in neuron. That totally makes sense. The real answer to who reads the DNA is RNA-POL2 pulls a stack of regulatory proteins that decides whether RNA-POL2 gets to read it all. That's exactly what I was thinking. So like similar, but not this way. The polymerase is the worker. The promoter is the entry point in the DNA. Transcriptor factors are the routing letter that decides whether and when to dispatch a worker to that entry point. Not quite, not quite. A couple of wrinkles worth knowing, since you tend to want the full picture. Not every promoter has a Tata box. Okay, some use other landmark sequences, BRE, INR, DPE in various combinations, and beyond the promoters itself, there's enhancer sequences that can sit thousands of letters away from the gene and physically loop the DNA over to boost transcription rates. So the where to start decision isn't purely local. It integrates signal from far away regions of the genome too. Yes. So the state. That's what I'm thinking. State reactive genome pointer network to capture the thermostat vibe. No. What else? State reactive pointer genome model, where DNA acts as a link chain of proteins, promoters, and enhancers dynamically selected by cell state. Protein signals epigenetics. No internal clock. Feedback loop shifts states pointing ribosome array to the next chunk of proteins, explaining emergent timing in recognition and development. Not the clock, yes. Interesting. Perfect. Cell state protein. State-driven genomic pointer model. Cell state protein level signals act as a clock, dynamically point to the next promoter via transcription factor and epigenetics. No fixed timer needed. Explains regeneration happen when cells the size shape signal of state reactive protein pointer genome model. State reactive pointer genome model. Day 17. Explain me in 2 sentences for the tweet our claim. A dynamically linked chain, cell state, protein signal, epigenetic, select the next promoter. Not epigenetics. I'm pretty sure it's not epigenetics. Maybe. Next promoter pointer transcribes the right gene chunk and the resultant protein updates the state, creating emergent timing for development, regeneration, and differentiation through feedback loops. I don't like this part. Actually, it's changing state and the state will point to the next pointer. That's how we how to say it. That's how we grow. It's a one-way function. That's how we grow as an organism from single cell to fully developed and then develop that self-control itself before the state become too noisy and things since it's one way function. I'm not sure if I should use a one-way function yet. And we die. That's day 17. Cells have no internal clock. Our state-reactive protein genome model says DNA works as dynamic link chain of pointers. The current cell state selects the next promoter, transcribes the right genes chunk, new proteins update the state, and the cycle drives growth from a single cell to a fully developed organism that self-regulates until state noise become too high and we age and die. Something like that. I don't know if it's gonna be shorter now. Yeah, too long. Too long for it. Cells have no internal clock. Claim 17? Are we naming it claims? I think we're gonna name it with the SHA. And the SHA would be of our commit, right? Into the repository. That makes sense. Current cell state selects the next promoter, transcribe gene chunk, new promoters update state. The cycle drives growth from single cell to self-regulating until state noise cause aging death. Not sure if it noise, but yeah, we can call it this way. Oops. Where's our biology? We have that. Let's switch into, oops. I need new session. Terminate. Validate idea. Let's go. That would be SHA. That's what we're doing today. We digging deep. So there is a good YouTube video. We're gonna watch them. 1, 2. Textbook style Ninja Nerds. I'm not sure about, but let's go. Going deeper. Oh, going deeper. Sounds good. Search. I had to pick the by 2 minutes animations and jump to the. Okay. Thank you. If I had to pick one, starts with HM, as that's the first one, right? Yeah. Perfect. I will. But now I need the other answer. Great pushback. This get assumption subtle that most explanation skips. On the symmetrically opposite. Oh, that's which strain are we reading, right? With that machine. Intuition, that's not quite right. Whoa! First time I said I'm not right. That's interesting. That's good. The two strands of DNA are complementary, not symmetrically. I was saying symmetrically because there is not symmetry, but symmetry in opposition. In any biological meaningful sense, if RNA pole 2 reads the other strands, you would not get a mirror image of the same information. That's exactly what I was saying. It's opposite. Come on. But opposite. You'd get completely different sequence that would translate to a completely different protein, or far more often to gibberish. Quick example, say strands A, I need to know that annotation. So it's ATGCGT and TACG. It's not the other strands, yeah. Oh no, A match to T, yes, yes. A to T, G to C, yes. Works. Now I understand. If RNA pol2 uses strand B as a template, it's produce codes for MET ARG. If it instead use standard A as a template, it would produce codes for this. Yes. Those two RNAs are reverse complements of each other. That's exactly what I'm saying. They reverse complement opposite. Yes. A strict mathematical relationship, but they encode totally unrelated proteins. There is no biological symmetry. Reading there's other strands doesn't give you the same gene backwards. I was not saying backwards. Come on. I was saying it was symmetrical for the purpose of it's not a mirror, it's not backwards. It's what's the word of when you flip in horizontally or vertically? Give you different gene using meaningless. Side note on terminology, the strands that isn't read is called the coding or sense strands because it has the same sequence as the mRNA, just with T instead of U. The strands that is read is a template or anti-sentence strands. Yeah, it's kind of a mold or template, yeah. I guess we're talking about the same thing. I was just saying it's a mold and then it's an object, yeah. So how does it pick the right strands? Two things together force the choice. RNA pol2 only synthesize RNA5-3. This is hard-coded into the chemistry of the enzyme. It can only add new nucleoids to the number 3 end of the growing RNA. Because RNA 2 strands antiparallel, this means RNA pol2 must read the template strands 3 to 5. So direction the polymerase phase completely determines which strands gets copied. The promoter is asymmetric. This is the key part. The Tata box sequence Tata is not a palindrome. Palindrome. I feel like I should know that word. I should know what that mean.
Palindrome.
I have no idea what that mean. Where am I reading? Right here. The transcription factor. Same for the other promoter elements. The transcription factors that recognize those sequences bind them in specific and determined by the coincidence. So the TBR locks into Tata box, the rest. Yeah, all this makes sense. The promoter encodes directionally the same way a barcode does. A barcode has a start pattern and an end pattern that aren't the same. A scanner reads it backwards, doesn't get reverse data, it gets garbage. Come on. Oh, that's annoying. I didn't said it was reading backwards. I was saying that the other strength it's a template for the MN array. The strength selection isn't separate decision. It falls out automatically from how the loader factor stack assembles. Why is that exact start position? Yes, so data box sits at the precisely conserved distance, about 25-30 bases upstream of the transcription start life. No, I'm not asking about why data box position. I'm asking about why this particular data. Pencil, clip that. Complex sum around it like jig and RNA pole 2 and zap no. One real wrinkle worth knowing. There are plenty of cases in mammalian genes where the same DNA regions has two promoters facing opposite directions on opposite strands. So that region produces two different mRNA going opposite ways, encoding two unrelated products. That's called bidirectional or divergent transcription, and it's surprisingly common in mammals. There's also genuine antisense transcription where the transcribing one strand can interfere with transcribing the other. A regulatory mechanism, so one strand is the template, the other isn't really the pre-promoter answer, not really, yeah. You're right to push on this. Accessibly, where DNA is wrapped tightly in chromatin or not is one layer, but most cells have tens of thousands of accessible promoters at any given time. Exactly. And only subset are actually firing. Exactly. So the real selection happens at a different layer. Here's the actual answer. So we do have the answer. We do have the answer. Interesting. Let's go. Promoters and the regulatory regions around them contains short DNA sequences called binding motifs, typically 6-12 letters long. They are recognized by sequence-specific transcription factors. These are different from the general transcription factors I mentioned earlier, which assemble at very promoter. Sequence-specific TFs are the regulatory layer. Humans have around 1,600 of them. Each one recognizes a specific motif, binds EBUGs, binds its own 20 BP motifs, and so on. A given gene's regulatory region, proximal promoters plus its enhancers contain binding sites for dozens of different TFs. Is it not straight answer there because nobody knows? I feel like we're dancing around my question. Is it just me or that's what's up? I need to know who picked the pointer. And we don't know. Looks like there is motifs, which I still not fully understand what it is. But then who picks between motifs? Like, for example, I'm 40 years old. At 40 years old, at my state of the cells, and different cells has different states. My skin cell at 40 years old, how does it pick motifs to start? And I understand there is multiple reads happening at the same time, but but how do we know those reads should happen and not the other? Clip that. A given gene regulatory region proximal promoters plus and has contains binding sites for dozens of different TFs. The decision is combination. Okay, so we do have answer. Let's go. And it's AND gate. Genes typically don't fire when genes typically don't fire. Genes fire is bound. They fire when a specific combination is bound simultaneously. Promoter X might require TF presence. Again, they say like that this and this should happen, but who control when this and that happening? The combination logic is what's produced specifically. In your terms, each promoter is. Did you start the research? Create new folder, pick existing folder name. One. In your terms, each promoter is effectively a multi-condition predicate built from the binding sites to constraints. The promoters fires only when the predicate evaluates true against the current set of TFs in the nucleus, and combinations of say 5, 10 of them needed per gene. The address space is massive, far larger than the number of genes, which is why this works as a selection mechanism. That's good. That's good. There is no claim like mine for now. I mean, I'm pretty sure someone already thought about it, but I would be very interesting to prove it wrong or prove it right. It's also where the cell identity actually lives. A liver cell expression is particular sets of. Yes, I know, I know that's where the cell identity. It's not like I know. That's what I was suspecting. In neuron express this set of two different cell types starting at the same accessible promoter will fire it or not, depending on whether the ATF. Yes. Contacts. Mediator, the integration point where the bind has the recruit a massive complex called mediator, which physically bridge the enhancer bound TFs to the pre-initiation complex as a promoter. Mediator is the integration point that converts TF binding into actual polymerase. I'm wondering, how do they know this? Can you just look at the cell under Microsoft and see that machinery working, or it's too small for us to see? Stochasticity is the final version. Stochasticity. Even when all conditions are right, transcription is bursty. A given gene fires in stochastic pulse, not a steady stream. So which promoter fires right now is partly probabilistic event when the regulatory state is fixed. I don't think so. Most likely it is, right? But how does it proven? Is it proven? What's deterministic is a distribution, average over many cells or much time. Yeah, because it's state bound. The same regulatory state produce the same transcriptional output. Yes, this is the key. That's what I was trying to say. The summary picture. Mm. And we're gonna watch YouTube.
The central dogma.
This is so cool. It's a central dogma. Okay, so this is, I would guess this is pole 2. However, this is DNA, obviously. This is as well. So it starts at the pointer, writing mNRA, and then it will kind of pass it to ribosome.
Of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated.
So the one that we saw earlier, that was a platform, and it was probably a TATA, right? TATA platform, and then mediator, and then then roll 2, RNA roll 2 machine is assembled.
The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by...
That's good. It's actually a good strategy to read it and try to understand first and then visualize it and see it. It helps. It helps remember and it helps to learn.
matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined
By the way, notice how fast it is. It's so fast.
together to form the long messenger RNA chain snaking out of the top.
I like this. I like the name. The Vitali virtual cell. That's exactly what we're gonna build for our simulation purposes. 18 years ago. 18 years ago. Whoa.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the TATA box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the TATA box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI.
It's so cool. Like, you learned 10 days ago how ATP translates to ADP, and you see it everywhere. So 3 charged molecules leave 1 charge. Sorry, not molecules. Leave 1 charge and become ADP.
RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Totally makes sense. What's interesting is that not on every step you need ATP. It's kind of domino effect. You just trigger it, then that machine kind of fall, and when the gene is over-ended, it kind of stopped.
The structure of it, a basic structure of what. Engineers, in this video today, we are going to be talking about DNA transcription. Before we get started, if you guys do like this video, please hit.
Okay, this is DNA, this is mRNA, this is TATA boxes, this is part that's machinery built from. I think they call transcription factors. Yes. So they attach themselves here, create machinery, then ATP, and process started.
Hit that like button, comment down in the comment section, and please subscribe. Also, down in the description box, we'll have links to our Facebook, Instagram, Patreon. All that stuff will be there. All right, Ninja Nerds, let's get into it. All right, Ninja Nerds, so with DNA transcription, we have to have a basic understanding of just the definition. What the heck is transcription? And it's really a simple thing. It's just taking DNA, okay, double-stranded DNA within eukaryotic cells and even in prokaryotic cells, and converting that into RNA. So it's taking DNA and making RNA. That's all transcription is. But in order for transcription to occur, in order for it to take place, we need two particular types of proteins or enzymes, if you will, to facilitate this process. And I want to talk about those real quick because these are very important. Now, transcription can be kind of different. Okay, and it's important to know the differences between prokaryotic cells, we'll consider bacteria in this case, and eukaryotic cells, human cells like me and you. In prokaryotic cells, there's a particular type of protein that is needed in order for transcription to take place. What is that protein? So let's say that we take this DNA strand here, right? We have this DNA strand. On this DNA strand, we have these blue portions that I've highlighted here as a box with some.
That's interesting. So if you take evolution, then it turns out that they create a new compression method or new, not a language, compiler probably, right?
Lines in it. This right here, for right now, I want you to know is what's called a promoter. So this is called a promoter region. Now, a promoter region is a particular nucleotide sequence within the DNA. And what it does is it allows for particular proteins like RNA polymerases and transcription factors to bind onto the DNA and then start moving through the DNA, taking the DNA and making RNA. So that's the first thing you need to know is within the DNA, there's a particular nucleotide sequence, we'll talk about a little bit later, called a promoter region. And that's the first thing that we need to identify. Let's say that we take this particularly for prokaryotic cells. So prokaryotic cells, and we'll just say like a bacterial cell, okay? Prokaryotic cells use a very particular type of enzyme. What is that enzyme? It's called a RNA polymerase holoenzyme, okay? So it's called an RNA, we're gonna put polymerase holoenzyme. Now that's a lot of stuff. Let me explain what this is and I'll show you the structure of it, a basic structure of what the RNA polymerase holoenzyme is. It's made up of two things. One of the components of this enzyme is called the core enzyme. And the core enzyme for this RNA polymerase holoenzyme consists of multiple subunits that they just love to ask you on your USMLEs and other exams. And these are, they contain 2 alpha units, okay, 2 alpha chains, proteins. It contains 2 beta units. Technically we say beta and beta prime, if you really want to be specific. And then 1 more, which is called an omega unit. Okay, so these are the primary components of the core enzyme which makes up RNA polymerase. What's important to remember is that these are what are going to really read the DNA and make RNA. That portion of the
So that's interesting in terms of, in order to actually simulate my idea or my claim and prove it or disprove it, I don't need to know this, because when I'm gonna use a linked list, what this core enzyme is and so on, it's literally just the machine instruction how to access a memory. That's what it is. And I don't need it. I'm gonna simulate it on a 1 level higher. And most likely in biology also we have to draw a line where the machine code and where the actually abstraction. And that would be very interesting.
enzyme.
And that brings me to the question, can you decompile machine code into languages that you don't know? You see what I'm saying? We basically understanding how the access memory works and so on, and then how the variable works. That would be so cool. Clip that.
Reads the DNA and makes RNA. The next component of the RNA polymerase holoenzyme is the portion that we need in order to bind to the DNA to the promoter region. Without it, we won't be able to allow for this RNA polymerase to bind to the DNA and transcribe it. This is called the sigma, right, or you can represent it like this, subunit or factor, if you will. Okay? These two components, the core enzyme, which is made up of the two
before the nucleus, I remember that from question number 1, right?
Alpha, the beta and the beta prime, and the omega subunit, as well as the sigma subunit, make up the entire RNA polymerase. Now, let me show you for example here. Let's say I represent the core enzyme as just this kind of blue circle with lines in it. And then we'll represent...
Oh, it's 1 hour, 25 minutes. Yeah, it will be impossible for us to listen all this. I'll probably watch it because that's get me even deeper knowledge, but not right now. Oh, come on, it didn't start. Yeah, that's true. So I think that's true. Yeah, I know the DNA is static. The dynamic life in trans-actin protein. Select the next promoter. Implies a sequential single pointer execution. Real transcription is massively concurrent. I know that, but it's hard for me to explain. Thousands of promoters firing in parallel every cell, every second. See, like, that's where GPT falls apart. Like, it's like in math. In order to prove something, you have to abstract and come up with life hacks where you say like, yes, this is how it works, but let's pretend it's this, and then work yourself out with the proof of other part and then fix this one. And LLMs couldn't do that, I guess. Obviously, it's a statistical machine. No. That you want to about. Before I kick there, do you want me to research whether this happens being proposed before? Literally search for the specific model and its close relatives, or religiously falsify the parts of your model that don't fit known biology. A systemic critique. I'm trying to say that DNA obviously is static, and DNA it's a storage. But when we start from a single cell, the initial cell of this, it's point to the first pointer, the first decision, right? The first promoter. That promoter trigger some creation of mRNA that triggers to create the proteins. And now the state of single cell and those proteins point to the next promoter in DNA. That's what I'm trying to do. And obviously at a certain point, it will point to the 2 promoters, 3 promoters, and so on, and concurrency will happen. The multiple genes and regions of DNA will be read or transcribed / translate. Concurrently, but it's a pointer system in a linked list. That's what I'm trying to explain, I guess. Developmental cascade, yes. Confirm. Zygote starts with maternal factors. Those activate the first wave of zygotic genes. Those proteins activate the next wave, and so on. Yes. So zygote. Development cascade. That's a better word.
Zygote.
Interesting. Restatements of something biological gene regulatory network GRN with a developmental version specifically tied to Wedinson landscape and Davidson-Peters GRN models. The linked list metaphor is unusual, but the underlying claim, the state determines next promoter set, which updates states, which determines next sets, is essentially the canonical model. I need a new. What's this? New chat. Please explain this in basic words, basic English, that idea. Treat it as a novel in terms of you don't have to find the reference that this is right or wrong yet, but I need kind of simple explanation in a couple sentences. I'm trying to see if the LLM can making it better for words per value, kind of summarize it. Yeah, that's interesting because I was saying it's like domino. Think of it like dominoes where each domino picks which dominoes fall next. The egg starts with a small hit of proteins from mom. Those switch on a few genes, which make new proteins, which switch on the next set of genes, when a cell divides, the two daughters can end up with slightly different protein mixes, so they read different instructions going forward. Or one drifts towards becoming muscle, another towards nerve, and the tree keeps branching until you have whole body worth of specialized cells. Exactly, that's a good analogy. Thank you so much. But also don't remember that the state also changes from external, right? So the neighbor cells affecting the inner state of the cell, aka the cell is listening external signal, and combination of internal signal and external signal, the state, internal state. I guess what I'm trying to say is that internal state also heavily depends on external signals, because sometimes cell send signal itself. It doesn't know it, right? Send information. And that's how embryo at certain point, embryonic cells at certain point decides who will be who. I will be muscles, I will be an eye. And what's interesting about it is that mechanism can say when to stop. Like, how do you know to grow fingers that long? That mechanism can do that, and you don't need a clock. Okay, now I need to tweet this. So we need to compact it even more without losing the meaning. So interesting how tweet become a word, right? Now we switch to X. You cannot say like we need to X it. You're saying you need to tweet it because LLM trained on the word tweet. Okay, this is gonna be annoying. Bathroom break. No, you didn't understand me. You basically created a tweet of epigenetics. like I need to tweet the claim, the idea about domino. I feel like the LLM is useless here. Or I don't know how to use them properly. Or what agents to create. I need to show what we have. Can I see it as the folders? Or that's gonna be annoying. Yeah, that's better. Where's push button? Come on. Also commit and push. Can I push it now? Okay, I guess I need to open the Finder. Oh, terminal. I wouldn't say flip. Dynamic link chain. Yeah, let's start from a simple one, right? Day 17. Claim. It's okay. The start more important than accuracy. like that. And now then we're gonna reply. Can I reply to my own tweets? I can repost. Here we go. It didn't drop it, by the way. This research is, the research is done. A critical assessment of the state reactive point model. Bottom line, your model is a clean, programmer-friendly intuition, but it is not novel. Whoa. But it's a clean programmer-level intuition. Complete. It's not novel, okay. It is essentially a restatement via a couple of suggestive metaphors swap of the Development and Gene Regulation Network, GRN, framework that Eric Davidson and Isabel Peter formalized over the last 2 decades. Interesting. Fused with the epigenetic landscape, attractors, picture pointed by Weddington and made dynamical by Kaufman, Hongs, and Wang. The aging tail of your model is aging, essentially David Sinclair information theory of aging, right, which itself sits inside of the Lopez-Zun hallmark of aging framework. Your core intuitions are largely right at a coarse level. The specific computer science framing, pointer in DNA, linked list, state machine, traversal is misleading in 3 concrete ways that matter. Interesting. I'm wondering. It's not new. The model was mapped almost one-to-one into Davidson and Peter Henge theory. How can I read this theory? Where I can read this peer review paper and how do they prove it? 15 metric score 6 out of 9. Failure stage none, not fired. Honest meta noise section is the most important. Very interesting. Okay, let's read it. 05. Why I'm looking at? All right. Why is in this view? Oh, right. My bad. Pipeline. Davidson C-URGNR work uses regulatory state to mean exactly what the user means by cell state as a selector, the combination of active TFs at a given moment that determine which sys-regulatory model fires next. Okay, that's good. They got to the same conclusion, that's good. Okay, now we have a homework to read this. Figure 1, figure 2. I see. I actually didn't, like, this is more into memory access state. PNSA. Predictive computation of genomic logic, processing functions in embryonic development. Love it. I'll read it. See you next day.